Hello, good afternoon, everyone. So my internet is a little bit wonky right now, so I'm not gonna turn on my camera. And if I start to sound a little bit choppy or if it becomes laggy, please let me know, and I'll see what I can do about it. All right. This week we are going to be doing lecture 7, which is protein synthesis. So let's get started. First question, what is the central dogma of biology? We talked about this several times before. It's basically asking you, what is the central theme of cell biology? So we start with DNA. And we are going to copy a section of the DNA in the form of RNA. And then we're going to use the information in the RNA to make proteins. So the process of making a copy of the DNA in the form of RNA is called transcription, which happens in the nucleus. And the process of decoding the RNA to make proteins, we call that translation, which happens in the cytoplasm. Okay, so maybe this happens at the rough endoplasmic reticulum. Maybe it just happens in the cytoplasm with those freely floating ribosomes. Okay, so that is the central dogma of cell biology. Next, we are going to compare DNA and RNA. This should be just a review. We've done this in lecture 2. So for DNA, it is going to be double-stranded, whereas RNA is single-stranded. The type of sugar for DNA, it is called the deoxyribose. So it is missing an oxygen compared to the ribose sugar, oxyribose in RNA. Okay, or just ribose. The nitrogenous base found in DNA can be A, C, T, or G. In RNA, we have A, C, U, and G, okay, where the U is the uracil. Okay. Different types of DNA. Well, there's only one type, just DNA. But for RNA, there are 3 types: rRNA, tRNA, and mRNA. Okay. Let's take a look at each one of these. First, we have the rRNA. The R stands for ribosomal. Okay. What does rRNA do? rRNA, it makes up part of the ribosome. Okay. Which is important for linking the correct amino acids in the proper sequence. Okay, so that's what the rRNA does, okay, during translation. All right. Next up, we have tRNA. The T stands for transfer. Does anybody want to tell me what the tRNA does? tRNA, what does it do? Okay, so Safi said moves from nucleus to ribosomes. Cynthia says brings the proper amino acid. Yeah, brings the proper amino acid is correct. So the tRNA will bring the correct amino acid to the ribosome. Okay. The anticodon, we'll talk more about that later, on the tRNA is complementary to the codon on mRNA. Okay, last but not least, we have the mRNA, which is the messenger RNA. Okay, so the messenger RNA is a copy of a gene, right? Which contains the instruction to make a protein. Okay. Any questions about the 3 types of RNA and what they do, where do you find them, and why they're important? Any questions about that? Okay, no question. Let's move on. List the 3 steps of transcription. Okay, so the 3 steps of transcriptions are as follow. We have initiation, elongation, and termination. So let me take a moment and explain to you what all these means. Okay, this is very important, so make sure you pay attention. Okay, so first of all, let's just help you understand where things are. So this is a chromosome, right? Chromosome. Okay, and then if I zoom in on the chromosome, that's basically a bunch of DNA, right? DNA like this. Ugh. It's hard to draw. It's basically a bunch of DNA, right? Like that. Chromosome is made up of DNA. So what exactly is DNA anyways, right? Well, DNA is just a bunch of letters, right? So this is DNA. So like, what exactly is this? Well, that is basically like A, T, C, C, G, C, A, C, G, A, C, just a bunch of codes, right? Okay. So DNA is double-stranded and it's complementary. So if I know one side, I am going to be able to create the other side as well. Okay. A is going to pair with T, and then C is going to pair with G. Okay? You don't really have to copy this down. You can just, sit back and watch. So the entire chromosome from like this end to this end is nothing more than a bunch of letters, right? All these letters. Okay. So if I take a section of the chromosome, okay, like this section, for example, right? That is going to be a gene. Okay. So a gene basically is a bunch of letters, okay? But I'm just going to draw it out like this, okay, like a blue ribbon. Okay, if you zoom in on this blue ribbon, then you're going to see all these letters. Okay, this gene is going to have three parts. There is going to be a beginning part, okay, which we call the promoter. Okay, the promoter marks the beginning of a gene. Okay. Usually it is rich in which nucleotides are we going to have a lot of in the promoter? Does anybody know? Remember from the lecture perhaps? Yep, A and T. That's correct, Savi. Rich in A's and T's, because A's and T's only have two hydrogen bonds holding them together. It's much easier to pull apart compared to C and G. And you need to pull the strands apart in order for us to copy it. Okay, so there's a promoter sequence at the beginning, and there is also the terminator sequence at the end. Terminator. Okay, which marks, oh, sorry, I had a spelling mistake here. Marks the beginning. And this one marks the end of a gene. Okay. So again, I'm just gonna maybe copy the letters here so you can see it, right? So like the promoter here might be like A, C, A, T, 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 G, T, A. Lots of A's and T's. And then the middle section here, right? Maybe I'll just copy what I have up there. Okay, something like that. And then you will have like a bunch of other sequence or something. Okay, it doesn't really matter, but I'm just showing you that that's what a gene would be. A real gene would be much longer. Okay, later on in another lecture, I'll show you where to look up genes on the internet if you're ever interested. So that's what a gene is. Okay, so I'm gonna have to make a copy of this. Okay, now DNA is double-stranded, RNA is only single-stranded. So which one am I going to copy? Am I going to copy the top one, or am I going to copy the bottom one? Okay, so that's the question, right? Well, it turns out of the two strands of DNA, okay, one strand is going to be called a coding strand. The other is going to be called a template strand. Okay, so let's say this one is the coding strand, and you cannot really tell. Like I have to tell you which one it is. The coding strand. And then the bottom one will be the template strand. Okay. The coding strand is the strand that is going to contain the actual instruction to make the protein. That's the one you wanna copy. Okay, but in order to copy that one, you will have to use the template strand to build your RNA. Okay, because when you build the RNA, it follows complementary rules. So here is a picture from your lecture notes. Let me just copy it over so you can see it. Give me 1 second. Right here. Okay, so here's a picture, right? So you can see over here, the blue is the DNA. Okay, and then in this picture, the top is the coding strand, the one that actually contains the information. And then the bottom here is template strand. So when the RNA polymerase, which is the enzyme that makes the RNA, comes along, it will read the template strand and put the complementary RNA nucleotide on. So it will see a T, so it knows to put an A. It sees an A, So it knows to put, supposedly to put T, right? But then because we're building RNA, we don't have thymine available. So we're going to use uracil instead. And then G and C and so on and so forth. So what you end up doing is your newly made RNA is going to be identical to the coding strand. It preserves the instruction, it preserves the code. And so you can see CAT here, I get CAU, right? So only T are replaced by U. Does everybody understand the difference between the coding strand and the template strand at this point? Any question before we move on? No, everyone is good. All right. So draw and label various part of a pre-mRNA. So the pre-RNA is the one that you would get when you copy your DNA, okay, before you process it. And so again, I'll draw this one. This is going to be your pre-RNA, and then it's going to have what we call exons and introns. Okay, so these part here, the blue parts, these are going to be called exons. Okay, so exons are coding portion of the gene. Okay, and then these ones here, the pink ones, these are introns. Introns are non-coding region, non-coding portion of the gene. Right? So what's going to happen is we're going to have to do post-transcriptional modifications. Okay, so in post-transcriptional modification, we are going to remove all the introns and we're going to join together just the exons. Okay, so like this. So that's called RNA splicing. Splicing. Okay. Remove introns and connect exons. Okay. And we're also going to add something to the 5' end and the 3' end. Okay, so here at the 5' end, you're going to add a guanine cap, 5' guanine cap, and then on the 3' end, we're going to add a poly A tail. Okay, many A's. Poly A tail. So together, the 3' cap and the 5' cap and the 3' tail will help stabilize the messenger RNA in the cytoplasm. So it's not going to like break down or something like that. Okay. So that is what happens in post-transcriptional modification. Okay, moving on. Complete the table below. So here's the coding strand. And we are going to make an mRNA from it. Okay. So remember, coding strand and mRNA are basically identical, right? Identical. But U replaces T. Okay. Because on RNA, we do not have thymine. We have uracil instead. Okay. So over here, if the coding strand is ATG, then the mRNA is going to be AUG. Okay. ATA is going to be AUA. We basically just copy everything but every time you see a T, you replace it with a U. Okay. So here, if I have the AUG, AUA, CGA, AAU, so on and so forth, right? This is the mRNA, right? What is the first amino acid, right? What does AUG code for? This is a codon, right? What does the AUG code for? So you will need to have your codon sheet, right? Okay. Now you must have a codon sheet for the quiz next Tuesday, because you want to be able to look up the codes. Okay. So print one out, have one on your phone. It's probably better if you have like a physical copy, but if you don't have a printer, then have it on your phone or on a screen. Okay. You will need to refer to it. So AUG, right? Does anybody not know how to use this chart? And now is the time to let me know. If everybody knows how to use this chart, I'm not going to explain. Okay. So AUG is a codon. Do you guys understand what a codon is? Codon. Do I need to explain what a codon is? Yeah, a codon is a code. Okay, codon is made up of 3 nucleotides. Okay, so nucleotides are ACTG. Okay, so then once you convert the ACTG into an RNA, and it's like ACUG, right? So when I have a code here, AUG, this is called a codon. 3 nucleotides makes up a codon. Okay, so there are a total of 46 possible codons. The first slot I can put, there are 4 possibilities, right? It can either be a C, a U, or a G. So 4 possibility. Same thing for the next one. Next one can also be a C, U, or G. And then same thing for the last one. So 4 times 4 times 4 gives you 64 possible codons. Okay, so the 64 possible codons are all listed here. Okay, so each box has like four codes, and then you have 16 boxes here in total. So multiply them together, you get 64 possible codons. So one codon codes for one amino acid. Okay, one codon codes for one amino acid. An amino acid is what makes up the protein, right? You should probably know that by now. Okay, so the situation here is we have to look up this codon to see what amino acid it codes for. Okay, so to use this chart, we're gonna look up the first nucleotide, so first base, which is A. So A is here, and then second one is U. Okay, so you see where these two intersect. So it intersects in this box, right? So I'm going to be focusing on this box. Okay, AU. So G, right? So the first one here is AUU, right? Next one is AUC, AUA, and then AUG. So AUG codes for the amino acid MET. Okay, methionine. Does that make sense? Okay, so just go back to, I'm going to copy this chart over. Go back over here. Again, you need this chart for the test. Okay, it's probably a little bit too small to see, but I'll make it bigger after I make my point. Okay, there we go. Okay, so for the first one, right? The first one is going to be AUG. And we just looked it up that it is going to code for MET, right? Methionine. Okay, so something has to bring the methionine over. Something has to bring that MET over. What is responsible for bringing the correct amino acid? What did we say on the first page? Anybody remember? Okay, the anticodon is on something. What is that something? All right. The tRNA is correct, Cynthia. Okay, so that's a tRNA. tRNA looks like this, okay? You just have to believe me. They do. And at the bottom of the tRNA, there is going to be an anticodon, okay? And if it's carrying MET, it's going to bring it to the AUG. But how does the body know that this is the right one? Well, the anticodon down here has to be complementary to the codon. So if the codon is AUG, then this is going to be UAC. All right. So if the anticodon and codons are complementary, then the tRNA is going to dock over here and it will bring the correct amino acid. Okay, so back to our situation here, right? If the codon is AUG, then the anticodon must be UAC. What does AUG code for? AUG codes for MET. Okay, let's do another one together. Next one. Next one is AUA. So the complementary anticodon is going to be UAU. And then we're going to look up what AUA codes for. Okay, so looking at our chart here, AUA, it is going to be ILE, okay, isoleucine. And then you're going to keep on doing it. Okay, so I'm going to finish this one for you. Basically, this entire row for the anticodon will be complementary to the mRNA. Okay, so GCU, UUA, CUA, AUU. Any question about how I'm filling in the anticodon row? You should ask if you're confused. Okay, amino acid, right? So we will keep on looking them up. CGA, right? CGA, which is arginine, ASN, ASP, and then stop. Okay, that is that. Okay, so please try the next one. Please try the next one. I'm going to give you a moment to do it. Okay, if you haven't already, please go ahead and do that. Okay, if you want, you can post a picture over here. I'll post a link on the chat box. If you have done it already, you can submit a picture, and then I can take a look at it. Okay. If you're done, you can also work on the next one. Okay. So I'm going to give you a couple minutes to do that. Okay. Anybody have the answer for this one that they want to share? No? Okay, I'll just write it out then. So for this one, this is the template strand. That means your mRNA is going to be complementary to it. So complementary to TAC is going to be AUG. Okay, CCC, GGA, UAU, GCG, and UAA. Is that okay? The anticodon will be complementary to the mRNA. So we will have UAC, GGG, CCU, AUA, and then CGC. Finally, AUU. And if you look up the amino acids, it will be MET, pro, gly, tyrosine, alanine, and then stop. Okay, so you can see there are 3 stop codon, which tells you to stop the process of translation. Okay. Any question about this one? Has everybody got the right answer for that? Okay. So I will post the answer later on. You can check the last one yourself. Okay, number 7. Given the following mRNA sequence, which 2 codes for the same protein? So what you have to do is you basically have to translate each of these sequence into the protein primary sequence, and you will compare them. Okay, so you can do that on your own with the chart, and you should find that the first one and the last one is going to be the same protein. Same protein. Okay. Next question. If a DNA sequence contains 360 nucleotides, how many amino acids do you expect to be in the protein sequence it encodes? Okay, so as I mentioned, 3 nucleotide makes up 1 codon, right? Makes up 1 codon, which goes for 1 amino acid. So to answer this question, we'll do 360 divided by 3, which gives you 120 amino acids. Okay. Assuming there are no introns in the sequence. Okay. All right, let's do some polls. Let's do this first one here. Go ahead. Okay, so only 3 of you answered. It is saying if the codon contains 4 nucleotides instead of 3, so there will be 4 blanks, and it will be 4 times 4 times 4 times 4, because each blank has 4 possible possibilities, right? So if you multiply all these together, you will get 256. So that is the answer. Okay. Here's another one. Okay. So this is the template DNA, and the corresponding sequence would be complementary to that. So you just use the base pairing rule, right? So T would pair with A, and then A would pair with U, and then so on and so forth. Okay, so this is the answer. Okay, let's continue then. Identify the type of mutation shown in each of the following. So this is the normal mRNA strand, and these are the amino acid that it codes for. Right? So we're gonna have to examine each of these and see what exactly is wrong with it. So first one, you can see the mutation happens right here. Let's use a different color right here. So instead of ACA, it is ACU. Right? This is called base pair substitution. You replace the amino acid. So the original codes for THR. This one also codes for THR. So because there's no change in the amino acid, no change, right, in protein sequence, this is called a silent mutation. Okay, or neutral mutation. Both are okay. Next one. we can see the change is here. So instead of ACA, it is CCA now, right? You replace the first base with C. So instead of threonine, it is now coding for proline. And this is going to change the protein sequence, right? We call this a missense mutation. Missense mutation. Okay, next one. By changing this and this base, you created a stop codon. And remember, there are 3 stop codon. If you look at the codon chart, right, the stop codons are UAC, UAG, and UAA. Okay, so those stop codon, if you introduce it prematurely, it will lead to like termination of translation. So basically, you will not be making the protein. So this is called a nonsense mutation. Okay, and you're going to create a shortened protein, or in this case, a truncated protein. We call it truncated. Okay. Over here, we have a base pair insertion or deletion. So if you take out one nucleotide, then what's going to happen is everything after that point is going to be wrong. So instead of reading it as like CCC and then ACA, because we took out the C here, it will be read as CCC and then ACA and then so on and so forth. So what happens is not only you're changing all the amino acid, you will also be missing the original stop codon. Okay, so this is a frame shift mutation. Frame shift because you are changing the reading frame. Frame shift mutation that will result in extensive missense. Okay, that means everything after the point of deletion will have the wrong amino acid now. This is an insertion mutation. So by inserting a nucleotide, inserting a base, you also create a frame shift. But this time, because the insertion caused a stop codon to appear, so this will create a nonsense mutation. So you can create a nonsense mutation by base substitution, or you can create a nonsense mutation by base pair insertion or deletion. If you delete a multiple of 3, right? So if 3 nucleotides are deleted, then no frame shift will occur. Okay, only lose 1 amino acid. Okay, if you lose 6 nucleotides, then you lose 2 amino acids. Okay, so it's less detrimental than a frame shift. Any question about the mutations? Okay. The following is a template DNA of a section of gene. Create base substitution that results in a neutral mutation. Okay, so we're going to need to have our codon chart again. Let me go grab it. Right here. Okay, so create a base substitution that results in a neutral mutation. Okay, so we can just choose any code, it doesn't really matter. Let's say we choose this one, AAT. So the mRNA is going to be UUA. Okay, so what does UUA code for? Well, we can come over here and see UUA codes for LEU, right? So we are looking for a neutral mutation, which means we don't want the amino acid to change. So UUA, if we look at the next one, UUG, they code for the same thing. So what you can do is you can just change the last one from A to G, right? Because UUA and UUG codes for the same thing. But we have to make the change on the original DNA sequence. Okay, so that means I have to change the T to a C. Is that okay? Please ask questions if you don't understand. All right. Can you do another one? Sure. So I can maybe pick this one. I can pick like, you know, CAG. So the mRNA is going to be GUC. So I'm going to come over here and look for GUC. GUC is valine, VAL. Okay. So we can see all these are all VAL, 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 right? So I can pick any one I want, right? GUC, I can change it to GUU, and it will still be the same thing. So I'm going to change this last one to a U. And when I go change it on my DNA, I have to make sure that it's complementary to U. So I'm going to put an A here. Is that okay? Okay. Next one, create a base substitution that results in a nonsense mutation. Nonsense mutation means you are going to create a stop codon. Okay, so let me write down all the stop codon. Okay, stop codon. Stop codon. Right? It's over here, right? All these are stop codon. So it's UGA, UAG, and UAA. Okay, these are the stop codons. Right? In the recording I said, right, you can remember it like this. Like, you got A in biology, you are good. You are awesome. Okay, so these are the stop codon. Now, what would the stop codon look like on the DNA? Right? What would the stop codon look like on the DNA? Okay, well, I am going to have to write a complementary. So UGA on the DNA is ACT. UAG on the DNA is going to be, it's going to be ATC. And then UAA on the DNA is going to be ATT. Does that make sense? I'm just writing out what the DNA would look like for the stop codons. Okay. So if I want to do a base substitution that would result in a nonsense mutation, then I look for something that I could change so that one of these things would show up, right? So for example, let me see, what's a good spot? Okay, so like over here, this is AAT. AAT. So if I mutate, if I change this one to a T, then it becomes ATT, which is one of the stop codon. Okay, so when you make the protein, you will make the first one, second one, third one, and then you would stop. The rest of it would not be made. So that's a truncated protein, a shortened protein. Is there another spot on this sequence where we can create a nonsense mutation? Please take a look and let me know. The last one, ATG into ATC. Yeah, you could do that. Okay. The first one, TAC. What are you going to change that to? Right. We're only going to do one change, right? One base substitution. So we can change one letter, make it stop. I don't think the first one works, right? Yeah. Is there another one? I see one more. CTC, yeah. What do you change CTC to? That's right. ATC. Are we together? Is everybody okay? I know I must have asked you guys this like 10 times every time, but I just want to make sure. All right, next one. Create an insertion mutation that results in a nonsense mutation. So I have to add a letter, a nucleotide, so that one of these things is going to show up. Right, so I'm going to see what's a good spot to add. So maybe here, right? If I put an A right here, then I'm going to have ATT. So that's going to create a stop codon, right? Make sense? Sometimes people have a misunderstanding. Okay, they're like, okay, what if I add it here? What if I add a C here? Then it's like ATC, okay? But that actually won't work, because if you do that, then this is how the ribosome will read it. It will read like TAC, GAG, CAG, GAG, AAT, and then if you put a C, then the next one it will read is CCT. Okay, because they read it in triplets, right? So if you put it in here, the ribosome is not going to read it as ATC, because the T belongs to the last triplet, right? Like the AAT, right? Okay, so that's why this will not work. All right. Okay, moving on. Create a base substitution that results in missense mutation. This is probably the easiest. Again, you can choose anyone. And let's say I choose this one, AAT. So that's going to be UUA. Okay, so if I look up UUA, it codes for leucine. Okay. But if I change it to, let's say UUU, then it's going to be something else different. So I scratch this, change it to U, which means I have to scratch this. That's going to be A. So that's going to be a missense mutation. All right. Okay, moving on. This is a sheet that you definitely should work through yourself. I'll do one examples with you, and then you know, the rest you can try and then check the answers on your own after. So we have a original DNA template sequence here. Sometimes it's useful to chop it up into like 3 triplets, okay, like this, so you don't lose track. And we're gonna make the RNA sequence because this is the template strand. So I'm gonna make it complementary. Okay, so UGG, AAC, CGC, UGC, UGA. So using the codon chart, which again you must print or have a copy of somewhere for the test, for the quiz next week. So that's going to be MET, TRP, ASN. Okay, these are amino acids, okay? CYS, and then stop. Okay. Now we have a mutated DNA sequence here. Again, it's useful to chop it up. Okay. Can anybody tell me, where is the mutation? Where is the mistake or the change? Second, yeah, the second code is wrong, right? So instead of ACC, right, it's ATC, right? That is the mutation. So if I copy the code, if I transcribe the code, it will be UAG, and then everything would be the same, right? Same as above. Okay. So what about the amino acid sequence? Well, AUG is going to be MET, just like before. And then UAG, that's a stop codon. Right? What kind of mutation is this? Well, you created a stop codon, so it is a nonsense mutation. Will it likely have an effect? Yes. Okay. It will likely have an effect. Will there likely be effects? Yeah. Yes. Okay. Because you're not making the protein. Okay? If you don't make the protein, you're not going to be able to use that protein. So this would be a very bad, very detrimental mutation. All right. So you can do it for all of them. Okay. So next one, I'll just start it for you, and then you can finish it yourself. Again, it's good to chop it up. And then you can see there is an extra nucleotide, right? Which suggests to you that something was added. Okay. And if you compare them letter by letter, you'll see this is being added. Okay. So that's an insertion. Okay. And then you will complete this and check the answer yourself. Okay. Moving on. Application questions. Deduce with reasons what process is shown in the micrograph. Okay. So the first one here, this is translation. Okay. You can see the green things that are coming out. Those are actually proteins. And then the pink one here in the middle, that's the mRNA. And then these little circle, these balls, those are the ribosomes. Okay, so this is what translation looks like. For real, this is like an actual under the microscope thing. This one, anybody know what this is? What are we looking at? What is this? What is this like thing over here? What are we looking at? That is the DNA unwinding. That is correct. So this is a replication bubble, okay? And we see replication bubble during DNA replication. Okay. And over here, this is transcription. Okay. That's RNA coming out. And then this is the DNA. All right. That is that. All right, let's do fill in the blanks. The process of copying a segment of DNA into RNA is called transcription. The process of using messenger RNA to produce protein is called translation. tRNA brings the correct amino acid to the ribosome during protein synthesis. Post-transcriptional process involves the addition of G-cap, guanine cap, on 5' end of the pre-mRNA and multiple adenines on the 3' end, called a poly-A tail. Something is the nucleic acid component of ribosome, rRNA. The anticodon is a triplet of nucleotides found in tRNA. It is complementary to the codon on messenger RNA. The start codon, which marks the beginning of the protein, is going to be AUG all the time. And then the stop codon is UGA, UAA, and UAG. The strand that is used to create the mRNA, that's the template strand. And then the coding strand is the strand that actually encodes for the amino acid. During splicing of post-transcriptional modification, introns are removed while exons are connected. Mutations are permanent changes in DNA. The promoter marks the beginning of a gene. It's rich in adenine and thymine. And the terminator sequence marks the end of a gene. Insertion or deletion of nucleotides that is not multiple of 3 can result in frame shift mutation. A nonsense mutation results in premature termination of translation. Okay, now let me ask you another question quickly. Here is the codon chart again. Let's just make sure that we are okay. And so this is the tRNA. This is the amino acid. Okay. If it's U, C, U here, what is the amino acid? Okay, we'll take a moment and think about it. You can type your answer in the chat box. Okay. Only one answer so far from Safi. Anyone else? The question is, what is the amino acid here? Okay, so you got to be careful, right? This is the tRNA. I am not looking up UCU, okay? Because this is the anticodon, right? The anticodon. So what would be the codon on the messenger RNA? If the anticodon is UCU, what is it on the messenger RNA? That's going to be AGA, right? So I actually have to look up AGA, which is arginine. Okay. Does that make sense? We do not look up the anticodon on the table. We always look up the codon. All right, so it's a trick question. Make sure you are okay with that on the test. Okay, that is it. Does anybody have any additional question? Remember, the quiz is next Tuesday. It's going to be based on lecture 6 and 7. 30 multiple choice in total. There's no application or multi-select this time again. And it's going to be 10% of your final grade. Okay. All right. Thank you for coming. Have a fantastic weekend. I'll see you all next week. Bye for now.